Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment&Health. Eco-Environment&Health is a journal initiated by prestigious scientists with a strong editorial board, including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design, and broadcasting. The experts will accompany you like an harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision-making that deeply integrates informatics, ecology, and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access feed will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future.
让汉森穿上鞋走了，我再说第几遍才能听得懂我说什么？
Hello, Veronica.
Hi.
Hi, Wei.
I'm Xie Zhiyu from Nanjing University. And now is the managing editor of E.
Excellent. Nice to meet you.
Mr. Yu, Professor Yu, your voice is so slow, so low.
就类似于说， Wait, can you hear me? Okay.
Yes, it's fine on my end.
Okay. Okay, I'm very sorry, Professor Gu Chen. He has another conference in Kunming, so he asked me to host today's talk. Hope you can understand.
Absolutely.
Okay, I think it's time to the talk. So, good morning and good evening, ladies and gentlemen. I'm very pleased to welcome you all to talk at EEH. It's a platform established by EEH, serving as a union of scientists sharing thoughts on eco-environment and health, especially in the time of pandemic. So thank you very much for your attention. I'm Xie Zhiyu from Nanjing University, now is the managing editor of the Eco-Environment and Health. Professor Morales and Professor Zhang, I think we have several sections in today's talk. So first I will introduce our general EEH, and then I will introduce today's speaker, Professor Morales, and then is the talk, and at last there is a question section. Okay. Okay. Now I would like to introduce a little about our new general EEH. It's called Eco-Environment and Health. It's a new open access general, aiming to be one of the most important flagship general in the fields of ecology, environmental science, and health. So the main topic of EEH include ecology and biodiversity conservation, environmental behaviors, and the bioprocess of emerging contaminants, human exposure and health effects, and the evaluation, management, and the regulation of environmental risks. So now I would like to take this chance to invite Professor Morales and Professor Zhang and all our audience to submit your manuscripts to Eco-Environment and Health. So this is a brief introduction of about our new journal. Now, today we are much honored to invite Professor Morales to this talk at EEH. It's my great pleasure to introduce Professor Morales. Veronica Morales is an associate professor in the Department of Civil and Environmental Engineering at University of California, Davis. Her work is driven by interest in discovering physical and chemical processes at the micro scale that control macro scale transport and the long-term fate of contaminants in soils and groundwater. She received her PhD from Cornell University and was a postdoc fellow at ETH Zurich. Thank you. So the theme of the talk today is groundwater flow and the mass transport in structurally complex porous media. So now let's welcome Professor Morales to give her talk. Thank you.
Thank you, Professor Yu and Professor Zhang, for the invitation to come give a talk in this exciting new journal. It's nice to be able to keep in touch. Professor Zhang and I did our PhDs together at Cornell University, so it's great to see a little bit of overlap still over the years. So I appreciate very much the invitation and very generous introduction. I would like to try to share my screen, but I don't have access to that.
Professor Yu, could you enable the sharing, the screen sharing?
I think you can share your screen directly.
No, it
No?
tells me that the host is disabled participant screen.
Okay, well. How about now?
Perfect. Thank you so much.
Okay.
Are we live?
Yes, I can see your screen.
Okay, excellent.
Thank you.
Well, thank you so much, and good morning to everybody. Or most of the folks, I guess, in the audience are morning time. We're here in California in the evening. But as Professor Yu was indicating, a lot of my research is motivated by understanding how water moves and how stuff in water moves, specifically when it's moving underground through rocks and soils. And how that stuff in the water, whether it's contaminants or something that we deliberately injected into the subsurface, how it moves, how it spreads, and specifically how it reacts when it's moving in confined spaces. Studying groundwater has been a really fun challenge for my career, kind of in general. It's a particularly difficult topic to study because the subsurface is deep and dark and largely inaccessible, which poses a lot of limitations as to how it is that you can study it, but it opens up an enormous door for creativity. And so with that perspective, I would like to present to you a couple of different ways in which we've managed to figure out a way to run experiments and run simulations that overcome a lot of the limitations of being able to study directly the processes that happen in porous media. So with that, and maybe I should get started. For those of you who have never heard of Davis before, and it's perfectly understandable, it's a very small town. It is located in Northern California, almost directly in the middle between San Francisco and Lake Tahoe, which is very famous for its very clean and beautiful waters. We are in the northern part of the Central Valley, which provides most of the food to the entire country. And as you can see, it's pretty far away from Los Angeles. So Disneyland is not a thing that we do on a regular basis. If anybody's around and manages to find their way through Davis, I would encourage you to contact me and I would be happy to show you around. And if you'd like to discuss some science, it would be my pleasure. Before I start talking about the research that we're doing, I'd like to just quickly acknowledge the people who have made the bulk of the work that I'm going to be presenting today possible. These are colleagues and students in particular who have worked pretty hard at various institutions where I've worked previously to realize some of the projects that I'll talk about today. Specifically, I'll be discussing the work of Zoe Canavas, who's one of my PhD students here at UC Davis, but also contributions from Fabian Arns and Maxence Farrell, who were both at ETH and UC Davis with me as well. Of course, I'd be remiss if I didn't mention the funding sources that are paying our bills. So the American Chemical Society, the National Science Foundation, and the AXA Research Fund. Okay, so I'm talking about porous media, in particular, the porous media that is important to study when we're referring to environmental systems. And if you're familiar with the critical zone, are the porous media that is the biggest and most influential pertains to soils and rocks. And the flow and transport processes that I'm specifically interested in better understanding are those that are related to groundwater. In general, flow is investigating the processes of the direction and the speed with which water is moving, whereas transport is referring to the stuff that is in that water, how that is spreading, how that is translating, and how it might be reacting with other physical, chemical, and even biological substances that it encounters along the way. Generally, when we're referring to groundwater pollution, there are three main culprits that provide sources for contamination. These might arise as a result of agricultural, industrial, or even urban activities. And in China, in the US, and in the rest of the world, we try to agree on a set of goals that allow us to continue using this vital natural resource. The first one being that we are trying to make designs in which way that the groundwater can be used more sustainably, that we try to do what we can to prevent any future pollution, even though there's a substantial amount already in place, and that we should do what we can to increase the success of any type of remediation strategies. So to get rid or at least mitigate existing pollution in natural systems. And even though these are nice and sort of apolitical goals, they really rely on two particular aspects that we often take for granted. The first one being that we already have a pretty decent understanding of the processes that are involved in how water is flowing and how contaminants are being transported inside porous media. And the second one being that there are already available models that are accurate for us to be able to predict how far this contamination extends. The point that I'd like to make is that we have essentially a very poor grasp of both of these aspects, which is one of the reasons why this topic is particularly challenging and requires an additional amount of effort and creativity put into place to be able to both understand what is going on, what are the processes driving groundwater flow and groundwater pollution, and also to be able to upgrade models that allow us to do a better job delineating the extent of contamination, but also allow us to better design remediation strategies. And so essentially, the problem boils down to the perspective in which we conceive how contamination is actually moving. And so if you were to take a typical course in groundwater hydrology and dealt with the topic of contamination, you might conceptualize that a plume of, the extent of a contaminant moving in a porous medium might acquire a shape that looks a little bit like this set of inscribed oblongs here. So if we're looking at a pollution that is moving with the direction of flow from an aerial view, we would find contours of concentration that approximately follow this shape, where the dark blue are the most concentrated areas, the lightest blue are the most diluted areas, and the signature is getting elongated as the flow is pulling it in the direction that it's moving. Now, if we were interested in figuring out what the concentration of that blue contaminant looked like at the location marked by the red X, we might find that the signal goes up and down in time at this particular location with a very predictable signature. So this is just a so-called breakthrough curve. It gives us the concentration at that location as it's traveling in time. And so initially, the pollution might have started here. It starts moving upward in the direction of the slide, and we would see the concentration at that particular location marked by the X rise up and then eventually come back down in a fairly predictable way. The signature would follow rules dictated by Fick's law, which really give us an idea of how things are moving according to concentration gradients. So, you know, the spreading would follow dilution, essentially. And this would be something that is fairly easily captured with mathematical models that assume that we're following normal statistics. So meaning that most things can be described by a Gaussian type of behavior. The irony, though, even though we call this normal transport or transport that is driven by concentration gradients and follows statistical rules that are Gaussian, is that hardly any subsurface environment or porous media is uniform enough, structurally uniform enough to acquire this kind of shape. The reality is that most signals are distorted, somewhat amorphous, And so, for instance, I'm showing here a picture, which is the cover of the National Research Council report from a few years ago of a contaminated site here in California at the beginning, and then 17 years after a pump and treat campaign. And what you can see is that neither of these two time points or maps of the pollution look oblong like the slide before. And so it begs the question of starting to ask what it is that is driving these amorphous shapes. And so the first thing to point out is that there are very, very strong signatures in natural environments where the concentration of, in this case, trichloroethylene is extremely high. These are created by so-called preferential pathways or preferential flow regions that allow the mass of contaminants to move almost undiluted. The consequence for that is that we get early breakthrough, or the signature moves faster than the average, it comes out sooner, and it's very undiluted. At the same time, there are these little pockets that, remember, the light blue is indicative of low concentrations. And there are these little pockets of flow stagnation, or where the water velocity, the groundwater is moving very, very slowly. And so it's receiving some of that contaminant by just molecular diffusion, and it's trapping that substance for extended periods of time. It takes a long time to get in there, but it takes even longer to get out. And so at that point, you end up with what in the concentration breakthrough curve that I showed earlier are very long tails, meaning that the concentration rises, but it decays very, very slowly and for a very long amount of time. The problem is that the combination of both having these preferential pathways and having these regions of flow stagnation gives us these very commonly observed skewed signatures of concentration that are no longer described by a Gaussian curve. We've seen this in a variety of settings, not exclusive to experimental sand columns that are brought into the lab. In very, very well-controlled field experiments like the MAID site in Mississippi, in cores that are extracted from the natural environment and brought into the lab for controlled experimentation, and even in entirely artificial systems, such as a sand bead or a glass bead pack that has been colonized with bacteria. And so no matter what it is that we do, we no longer see this Gaussian shape that rises and drops down pretty quickly. What we often see are these very long tails, the quick rise, the earlier the normal breakthrough, and the very slow decay at very long times for low concentrations that ends up being problematic, particularly when we're dealing with contaminants that are toxic at very, very low concentrations, because it means that you're going to have a continuous source, particularly from these regions of stagnant flow, that are continuing to taint the quality of that groundwater. And so it's important, and I think we're doing a pretty decent job for the field of contaminant hydrology, that there is more of a recognition that conventional models that assume that things are moving according to an oval shape, and most of the processes are driven by concentration gradients or Fick's laws, but they absolutely cannot quantify some of the typical signatures that we see in structure in porous media that is structurally much more complex, such as anything that pretty much we can find in real systems. And with this first recognition, there's been a push to have better and more advanced models that can capture some of these particular features. And people have been studying them at the core scale, and even at the field scale, where no matter which scale it is that you look at it, we see these heavy tails that absolutely cannot be captured by conventional models. And so as Professor Yu was indicating, my team is looking at the physical and some of the chemical processes that allow us to understand how small scale behavior can describe much larger phenomena. And so specifically what we do is use a probabilistic approach to describe what is happening within very small ranges in confined spaces that allow us to build statistical models that describe anomalous transport as the collection of millions of interactions between particles and individual pores, so that we can begin to capture these features of early breakthrough and late tailing that are pretty common in most structurally complex systems. Okay, so in many ways, this is complementary to the work that has been done in the past, but we are pushing the envelope a little bit further by continuing to study processes at the pore scale, but extracting the key statistical features that allow us to describe larger phenomena that are not limited to just a couple of millimeters in space. So with that introduction, I guess what I'd like to do is discuss two of somewhat recent projects that we've been working on. The first one being a way to understand how velocity within the pore spaces evolves both in time and space, and how that can be understood from the perspective of a particle moving through a porous medium. The point is to study, again, these statistical rules, the emerging behaviors or dynamics that allow us to understand what is going on at the pore scale, but to use those statistics to parameterize larger scale models as the collection of millions of these interactions between, individual molecules of a contaminant and the billions of pore spaces that are available that create the matrix of the porous medium that it's moving through. So with this approach, we are able to not just capture the average behavior, but also quantify some of the rare events that are responsible for the early breakthrough and the tailing that is so problematic for contaminant transport. So for this work, what we ended up doing is gathering data, empirical data, so experimental work, through the tracking of individual particle tracers moving through a transparent porous medium. By making the medium, which is indicated in the panel below, optically accessible, then we're able to move or use a camera to track the behavior of tracer particles, in our case, polystyrene beads, as they're moving through individual pore spaces in this three-dimensional sample. The particle trajectories are shown in the panel above, and they are color-coded by their local velocity. And being able to track them in space and time allows us to pretty much quantify anything that we care to know about their dynamics. And so rather than trying to use the conventional approach where we look at a concentration as a map of the concentration, but we're looking at it from the perspective of discretized particles. So rather than following the Eulerian framework, we're using a Lagrangian approach to be able to understand the exact same system. We are particularly interested in analyzing and better understanding the process of how velocity is changing both in time and space, because we believe that this holds a signature to be able to build better predictive models that tell us when things are going to come out earlier than the average, and for how long a very low concentration is going to persist, given the diversity of velocities that we can find in structurally complex systems. So for the sake of simplicity, I'm just going to talk about the results of this third sample. We created porous media of different levels of complexity to try to systematically understand what was going on. But the results essentially boil down to the same thing. So I'll just be talking about this third sample. And so to be able to collect these data, the very first thing that we needed to do was gain optical access. We know soils are opaque, and it's very difficult even to use microscopy to see inside of them. So what we ended up doing was making a fake soil that is completely transparent, or so-called index matched to the liquid. And so you can see a cell here that is fully drained. So it's just the grains in there. And the cell on the right-hand side has been wetted with the liquid and the grains, and we can see perfectly through it. So it's optically totally transparent. So once we have that set up, what we were able to do is use a technique called particle tracking velocimetry that allows us to track these individual tracers moving through the porous medium in 3D space and also in time. The technique is fairly simple. So one has to illuminate, this is the cell, that little cube that is shown in pictures below. It is illuminated with a coherent light source as one is injecting a fluid that has been seeded with tracer particles. In our case, we use fluorescent polystyrene beads. And we're recording the movement of those particles through the media as they're traveling through the pore spaces with a high-speed camera and an image splitter. The image splitter is particularly important because it allows us to image the sample from a couple of different angles. That gives us stereoscopic viewing of the sample and allows us to reconstruct the position of the particles in 3D. Subsequently, we took these structures that are totally transparent, and it's very difficult to tell what the structure looks like, but we put them in an X-ray computed tomography instrument that then takes a three-dimensional picture in grayscale of what the pore space looks like, okay? And so with this information, we are able to not just know how the particle dynamics are behaving, but we would also know information about the structure that is causing these dynamics. And so I guess we'll talk about a little bit of the results. So for starters, the first thing that we know, or what motivated this work, is that assuming that particles are moving all according to the same average velocity is one of the underlying assumptions of conventional models that we suspect is incorrect. And so the first thing to understand, now that we have detail about how these particles are moving, how quickly they're going, and in what direction, is to understand essentially the first two statistical moments of those particles. And so what I'm showing here on the left-hand side is the average velocity of the particles as it evolves in time. And so this is so-called the mean displacement. The first thing that we note is that the trend is quite linear, even in log space. And so this tells us where the center of those plumes is at every single time. The behavior of this particular statistic is not so interesting. It essentially just tells us that the average velocity is constant for very long periods of time. And so that's fine. We know that predicting the center of that concentration signal is predictable. What is not so predictable or straightforward is understanding how quickly it is spreading. And so the plot on the right-hand side is also showing the temporal evolution, but instead of the average, it's looking at the variance. So this is the so-called mean square displacement, and it tells us how quickly the spreading is occurring. So we know where the center is, but we need to also understand how quickly it's spreading. And this is not at all driven by concentration gradients, at least not at early times. If it were driven by concentration gradients, the scaling between the variance and the time would follow a slope of 1. But we can see here that it follows a slope of 2 and then a slope of 1.5 for an extended period of time. It eventually gets to 1, but not immediately. And so this is the first piece of signature that allows us to then recover the so-called statistics of the pore scale that are going to help us better build models that describe large-scale behavior. So this faster than concentration gradient rate is one of the hallmarks of anomalous transport that we are going to be using to improve our predictive models. The next thing that we needed to do is understand how long these particles are spending in these preferential pathways versus the stagnation zones, and how long it takes to transition between them. To be able to do that, we need to, first of all, understand the spatial correlation of those trajectories, how long it is that they're spending at that same velocity as a trajectory is traveling through space. So that is given by the autocorrelation function on the left-hand side. And once we understand how long these particles maintain that same velocity, the next thing to know is, well, what velocity are they even preserving? And so that is given by the probability density function of velocities on the right-hand side. So we know that there's going to be a bit of a memory that particles have of preserving a velocity that is being sampled from a log normal distribution, as illustrated here. And that they will transition into something different after some distance that they've traveled within the forest medium. And so this gives us now most of the parameters that we need to start building our transport model. And so the description that we are looking for specifically is an evolution of a velocity process that changes both in time and space. And so what we've done is now deconvoluted those statistics to understand how things are moving in space, but now along the pathway of those tracer particles. And we've been very fortunate to find stochastic processes that describe the behavior that we've seen in our system specifically. So number one, that the random velocities that are being sampled are coming from a log normal distribution. And the second one is that they are showing some level of spatial correlation, or so-called Markovian process. And so an Einstein-Uhlenbeck process is a well-known stochastic system that allows us to recover these two details of the particle dynamics. The first one being that the system is log normally distributed, and one can cheat in mathematics to turn a log normal distribution into a normal distribution by taking its natural log, as shown here in red. So if we wanted to predict the velocity of something by assuming that it contains properties that are Gaussian, then we just need to take the log of that to transform it. We already know the distribution parameters that describe the log normal distribution of the thing that we actually measured. So that is what is shown here in green. But because it's a stochastic process, then there's a level of noise that needs to be implemented. This yellow term allows us to incorporate a bit of that white noise that is, by definition, molecular diffusion, which we see in any system that is driven by water. And so essentially the way that the model is broken up is that instead of looking at how concentrations are changing in time and space, what we do is track discrete particles and build unique trajectories that follow equations of motion. Using Langevin equations in discretized space, essentially what we do is rebuild these trajectories very much in the same way that we measure them experimentally, but tracking them along the trajectory path. So this is the space equation, but also in time with this equation. And so briefly what this is saying is that we're going to be building individual trajectories where they get displaced one delta t at a time, so little jumps. And they're going to be jumping little distances in space as well, but the change in time is going to be driven according to the velocity that it happens to have sampled at that location. So this WM term comes from the stochastic process that I described just prior. And so by being able to build these individual trajectories, millions of those, we can recompose the plume behavior that we've seen before and do a better job at describing this anomalous transport. So number one, we're able to reproduce the velocity distribution very, very well. So here in cyan, what I'm showing are the data experimentally, and then black are the model results. And so we get pretty darn close to the behavior that we measured experimentally. For the displacements, both the average and the variance, as they're changing in time, we are able to reproduce this faster than Fickin or faster than concentration gradient spreading behavior really, really well. And lastly, because we're working with a model that describes the behavior of individual particles, we're able to reproduce this intermittent behavior, which is really a representation of having very long periods of low velocity that are interrupted by short bursts and high velocity. This is exactly what is happening within the pore scale, where you have pores that are doing this Coke bottle behavior, and the particle is accelerating, decelerating, accelerating, decelerating. And so even that kind of behavior is captured in velocity increments that are shown here on the far right. But the takeaway from this comparison is that the approach is extremely powerful in upscaling the statistics that we're able to gather from the pore scale, doing very small experiments, and describe behavior that is unique to much larger problems. I should say also that this is an extremely computationally efficient approach. And so even though it's numerical, we're able to get to the answer of how things are going to be spreading at large scales extremely efficiently. And we've been using this kind of model to work on more complex systems, like those where the porous medium is evolving. And so information about how when bacteria are living in soils or rock and are blocking individual pores, how that distorts and changes the behavior of how material is moving to porous medium. And we've also began revising some of the fundamental questions for filtration. So specifically for colloids, which are suspended matter that is moving around in porous media, but it has finite size. Okay, so I think moving right along, I'll talk now about the second project that we've been working on. And this one, we are kind of going back to revisiting the question of flow. So the first problem was dealing with transport. Now we're going back to flow and trying to understand what are the structural features that are causing preferential pathways to form. Why is the system showing very uniform and very easy to predict flow? So the velocity and the speed, the velocity and direction in which the parameter is moving, and why are other structures that seem to be very similar displaying very strong preferential pathways and very strong stagnant zones. And so this is the work that my students Fabian Arns and Zoe Canavas have been working on pretty hard during the pandemic on. And so we've been, because we're looking at the process from a pore scale, and they are very much interested in marrying information from other fields, they decided that graph theory was a pretty good approach to be able to represent a porous medium with variable resistivity between the channels. And so we gathered a rich set of granular material from the X-ray tomography that we gathered before. We had a bunch of different digital samples, solved numerically the flow field, and you can see that here it's listed by the local velocity and color-coded in these granular materials, and started to make assessments about how the structure is influencing the local velocity. And so the workflow for this particular project goes as follows. We took the three-dimensional images of our porous media, deconstructed them by the individual slice, solved the flow on the one hand, and analyzed the structure as a graph of the pore space on the other. And the first thing that we needed to do was establish a metric that allowed us to even quantify channelization or how non-uniform that flow is. So there is no established definition about when one sees or doesn't see preferential flow. And most people consider this to be a system that is binomial. So you either have preferential flow or you don't. The reality is that it's really a spectrum. And so you can have a level of channelization that is very pronounced and one that is very weak. And so we came up with a way to quantify that as indicated in the figure above. And so these two samples have a very similar porosity exactly the same grain size distribution. One of them, the one on the top, has a very uniform flow moving through it. So meaning that any pathway that you choose to take is going to be about as fast as the next one. The sample on the bottom, however, has a very pronounced preferential pathway marked here again in red, and very, very, very slow stagnation zones. And so by applying a percolation theory to the flow field, we're able to come up with a quantitative way to assess how pronounced these preferential pathways are. And so that's been a really nice way to get started on even quantifying this phenomenon. The next thing that we needed to do was assess the contrast between preferential pathways and stagnation zones. And so the plot here, shown on the bottom right, is trying to illustrate that exactly. The red lines correspond to the sample at the bottom, the blue lines correspond to the sample at the top, and the dashed versus the solid line indicate the difference in expected velocity if you were to be in the preferential pathway, so the solid line, or in the stagnation zone, which is the dashed line. And what we can see without a whole lot of surprise is that if you're in uniform flow, the difference of being inside or outside of the stagnation zone is minimal. However, if we are in a system that is seeing very pronounced preferential flow, we see a big difference between these two velocity PDFs. So knowing exactly where you start moving as a particle makes a huge difference if you're in this kind of structure versus this one where it actually doesn't matter at all. And so at this point, what we have is a pretty decent way to at least quantify this phenomenon of flow channelization. The next thing to do is come up with some information that allows us to use that to be able to predict where it is that these pathways are forming. And so considering that from a numerical perspective, the width of these channels dictates essentially Poisson flow, or how quickly that flow needs to move through this network of capillary tubes. We decided to parameterize a pore network model that is trying to bias the shortest path or find the minimum resistive path according to information about the structure alone, and having that match to the flow field that we already solved separately. And so what I'm showing here is 3 different models, and we tested hundreds of these with different information about the structure that would eventually give us an evolution that matches that of the displayed flow field. And so by finding a biased shortest path analysis, we're able to essentially overlap and identify which structural feature is the one that tells us exactly where the pathway is going to be forming according to the Navier-Stokes equations. And so we did this for the thousands of samples that we had to find the one parameter or the set of parameters that are structural that tell us exactly where it is that these pathways are forming. We did a few checks just to verify that this was not a fluke, that the structural information indeed gave us a good recovery of where the preferential pathways are forming according to our direct numerical simulations of the flow. And according to the control, which is shown here in blue, and the accuracy of our our model shown in orange, we see that we do a pretty decent job at describing that. And that particular structural information is, in fact, key to be able to say where it is that the pathways are forming. Now, the last question that we wanted to ask was, why are they forming there? And we suspected that this had to do with organization of the pore spaces. And so a porous medium can be thought of as a level of disorder in capillary tubes that are connected to each other, but they're not all disordered. And the way that they are disordered makes a big difference. And so because we're already using graph theory as an approach, we decided to dig in a little bit more in that field to find what sort of established metrics there are to reveal the level of order in the channels. And so we found one in particular that assesses exactly that kind of information, suspecting that the pathway is forming along these edges not because these are the fattest channels with lowest resistivity that are interconnected to each other, but they are the widest ones that are continuous within the porous medium. And so by using this assessment of assortativity of the pore network, we're able to assess how ordered or disordered channels are along the preferential pathway and compare them against the level of order or disorder, however it is that you choose to think about it, in the stagnation zone. And so the data are plotted here on the bottom right corner, where we see a metric of the level of order in the preferential pathway, what we're calling the primary flow subnetwork, against the y-axis, which shows the level of order in the stagnation zones. So everything else that is in blue. And what we can see is that most of the data fall below the 1-to-1 line here, which indicates that in order to be in a preferential pathway, your pore structure is very ordered. And we also see trends here that increase more strongly with how channelized the system is, or how much more preferential the system goes is redder. The more uniform the system is blue. And so this is really exciting because it allows us to now infer dynamic features from information about static properties of the structure alone. And so I think I've run out of time. I think I'll just summarize now quickly some of the key points about flow and transport in these structurally complex systems. Hopefully, if there's one thing to take away, is that signatures of transport in porous media are very anomalous. So generally what you see is a very early arrival of a substance and late tailing of low but significant concentrations. And this is really the norm rather than the exception in groundwater systems. For the first project that I presented, it's important to keep in mind that in order to correctly parameterize these models, we need to understand how the velocities are being sampled by particles, both in time and space, because that really is what controls how the plume is spreading. And they generally spread faster than was dictated by concentration gradients. And finally, what I wanted to say is that the arrangement of how the flow is structured within a porous medium really is dictated by the microstructure of the pores themselves, and how the channels are interconnected to each other plays a huge role in dictating whether you're going to have a preferential pathway or not, and where it is that it's going to be located. And so with that, I thank you for your attention, and I'm happy to entertain any questions that are that have come up. Thanks.
Thank you, Professor Morales. It's a very interesting and professional and excellent talk.
Thank you.
Thank you. Now it's a question time. So are there any questions for our audience? And in Zoom, you can raise your hand, and in other platform like Bilibili and Co-Share and YouTube, and you can write down your question, and our assistant will paste your question into our chat room. So can I ask the first question, Professor Morales?
Absolutely.
Yeah, I understand you build a new model, and it's a micro scale model, right?
Mm-hmm.
Yes. Yeah. Okay. So I'm wondering, can it be used in? I think it's can be used in many ways, like predicting the fate of some pollutants in the soil groundwater system, and also some other like oil recovery and geotechnologies. And so are there any examples it's applied in this area?
Yeah, sure. No, thank you for the question. Yes, so the short answer to that is yes. You can certainly use these kinds of models for describing a variety of pollutants. It's a fairly new field that is pulling information out of chaos to apply it to an environmental system. And so there are not well-established platforms in which one can just say, okay, I will, for instance, plug in the information about the retardation factor and I don't know, a degradation coefficient of said material and spit out properly the expected reactive transport from it. But it can capture some of these signatures of the lack of reactions in a variety of pollutants much better than conventional models and in a much more physical way than just saying that there's correcting factors that need to be tweaked in order to reproduce the signal that we see in experiments. Regarding your other part where you're discussing whether this can be used for oil recovery, I think the answer to that is also yes. I have not seen a whole lot of work on that in multi-phase systems, which is at least the area of the literature that I'm familiar with for oil recovery. But there's certainly a lot of work with the general community that I work with to try to make these models available for reactions and also for multi-phase systems.
Thank you.
Hello, Veronica. And it's okay. So I really enjoy your talk, and it's very nice to follow your progress on this area of research. I think it's very interesting, and it's very important. And I remember the days when we were in grad school, and we thought about this kind of a complex problem. I thought, gosh, I wouldn't be able to solve it by myself. And it's really great to see that you're making great progress in this area and on this very difficult problem. And I think it's very important. And one thing I really like it because when we think about the contaminant feed and transport, especially on some of the very challenging pollutants, some you may know like a PFAS, like a poor and polyfluoroalkyl substances. And now the regulation level or the proposed regulation level are very, very low. So if you have a contamination source, it's really important to understand that tailing of the transport. And so have you thought about apply like what do you learn in studying PFAS transport?
No, that's a great question. I've briefly thought about applying this to PFAS and been deterred from that very quickly because there's a variety of other interactions that need to be accounted for with that particular substance. So you know, I mean, wait, I've known you for 15, 20 years now. I like to study the fundamental processes. I'm less concerned about the specific contaminant that we are studying, because to me, the interesting part of science is to get to the root of what is driving a bunch of different materials. PFAS is relatively new. I think we don't entirely even understand how it interacts with organic matter and with other biological stuff that is underground. So it makes it particularly tricky to do that. But I am sure that somebody in my community is going to be working on PFAS at some point soon. But you're absolutely right that we are very concerned about capturing the main signal and often forget that these very low concentrations that are extended for a very long time are just as problematic as the highest levels, but they're very difficult to measure. One needs to have both the patience and the instrumentation that is required to get at them correctly. And so even just getting data to build these models has been challenging. So we're working on being able to understand that in suspensions a little bit better. And the application for that would be, to better understand when the prolonged signature that you get both with viruses and with materials moving in the environment. But PFAS would be a really, really great, really important application in emerging contaminants right now.
Yeah, the reason I was asking is because like you published a paper on organic matter, natural organic matter, and its interaction on colloid particles and its aggregation and transport behavior, right? Because PFAS have some property like of surfactants, and it does interact with air-water interface. So I thought like some of the setup in your lab seems make it very ideal to at least to focus on like very particular processes that may drive the PFAS transport. So we were founded with a couple of PFAS projects. We're very interested in looking for collaborators. So we may be able to join force again to tackle this problem.
That would be great. Yeah, let's do more about that.
Yeah. And I will see if other audience have more questions. And I do have a second question, but I will wait for my next turn.
Sure.
Thank you, Professor and Professor Morales. Are there any questions from the audience? And I think I have another question. Sorry. Actually, I do some research about soil remediation, and also some risk assessment. So we used to use some larger model like SWOT and HydroRAS, and to do some risk assessment about the non-point source in soil water system. So I'm wondering, can your model be integrated into the larger model like SWOT and HydroRAS?
With HydroRAS?
Yeah.
So it's different than HydroRAS. It is an alternative to the governing equations that are implemented in HydroRAS, because so one of the things that I tried to explain, and maybe it wasn't so clear, is the perspective with which you analyze this plume concentration is fundamentally different in this approach versus the approach that HydroRAS takes. So from HydroRAS, you look at how a concentration is changing as a function of time and space. So generally, you're looking at partial derivatives of concentration as a function of advection and dispersion, giving those as constant terms, maybe some reactions. Whereas in this particular process, advection is not really a term. We're looking at the advective diffusive equation instead of the advective dispersive equation. And so everything is very local. The advective term is not the average of everything. It is the actual local concentration, the local velocity that you experience moving in this complex system, which fundamentally cannot be represented by one average term. Instead, you must describe it as a probabilistic function instead. And so it would be, I think, not necessarily possible to marry these two approaches. I see them more as alternatives to describe the same kind of behavior.
Thank you.
Okay, I guess I would go ahead to ask my second question. I think in your presentation you mentioned about using graphic theory to describe or understand some of the patterns and the flow patterns and the behaviors. Right. So have you thought about using other like a machine learning method to study it? The reason I was intrigued by this question is because I think in general we thought a machine learning method are opposite to like a mechanistic driven method, right? Machine learning does not really tell us. Well, I probably frame it wrong. I guess what you show is that you can use machine learning to understand some basic processes.
Mhm.
Is that right?
Yeah. So the way that I think of machine learning, and this is actually using some level of machine learning to identify the structural feature that matters the most. That actually gives us the match between structure, the lowest resistivity path based on only information about the structure, and resistivity according to the solution to the Navier-Stokes equation, which is as detailed as you can get.
Right.
The reason why we opted for more physical representation is because we wanted, as you said, to understand the mechanism behind that. So we wanted to know exactly which feature mattered instead of just a convoluted and potentially black box equation that gives us the right answer without necessarily having a fundamental understanding of why that is the right answer. So for that particular project, I think you very nicely picked up that we're interested in answering what is the cause for preferential pathways, how are they formed, and why. And these are three things that you need to understand with a mechanistic approach and not just a black box approach, which is what maybe deep neural networks would give you. They would give you the right answer. They're going to be forming according in this particular location, but you may not have the luxury of saying why that happened, which for us was really important to be able to discern.
Okay. So some machine learning method will be able to pick up some important features among a vast amount of data. So there could be some way like using that as a tool to understand. So that does enlighten me, because previously I thought machine learning is, like you said, a black box approach, but it seems that we could use the machine learning to actually understand what's inside of the black box, even though we're using a black box approach.
Yeah.
All right.
And it can often lead to very, very high accuracy predictions, which is all, I mean, of course, it's desirable to get the right answer. For our group, it was important not just to get the right answer or as close to the right answer, even if it comes at the expense of understanding why. So the why that happens was really crucial. And we started initially with machine learning approaches. We didn't like the answer that we were getting because it was impossible to interpret. So we decided to go back to a little bit more rudimentary approaches and still get an acceptably good accuracy. But I know there's quite a lot of work at UT, at the University of Texas, that is working on that, particularly with Masha Prvanovic is definitely working really hard on that front.
Okay, I think there is a question in the chat box.
The question is, what about the applicability about the model? What do we need to pay attention to in the application of the model? The applicability. I think maybe this question is asking where one can use it and where it might not be suitable. So for the type of model that I presented here, the applicability is limited to conservative transport, meaning that it will describe correctly how the particular contaminant plume is moving on average and how quickly it is spreading. So this works for a variety of substances that are not interacting with a rock, meaning undergoing equilibrium absorption and not either reproducing or getting destroyed. It can be amended to account for these features, but the way in which I presented it for this particular project, it was not dealing with any type of reactions yet. However, it begins to describe mixing, which is a precursor for reactions that is extremely important and underappreciated in most conventional models that deal with some level of destruction or addition or let's say, reproduction of a contaminant. And so this would be where it is that I would say is important to pay attention to the applicability of the model. Is that if it needs to account for reactions, there's an additional amendment to use, but at its core, it describes conservative transport.
If I could, and I would like to ask the third and the final question, because this is really intriguing me, and the preferential flow, the formation of the preferential flow. Veronica, you probably remember when we were in grad school, we became aware of the experiment Thomas Steenhaus and Yves Palange did in 1980s with the sandbox experiment, right? Like I think you also show with the glass beads that when you have a rather homogeneous materials and you will have I would say probably the persistent preferential flow pathways. And it appears that to me is once this like a pore structure is set, and then you will have, is that right? Like a persistent of preferential flow pathways that wouldn't change, but once the structure is disturbed or change, like physical structure, the pore network is disturbed, and then the pathways for preferential flow is going to change, right?
Yeah. So that's definitely the case.
Okay.
Yeah, so we're studying that in the context of bioclogging and also for dissolution. And so you're in time constantly changing the underlying pore network, which means that the path of least resistance is also changing in space as the process is moving forward in time. And so these pathways are not going to be steady. And it's important to understand where it is that they're evolving, if the ultimate goal is to be able to understand, let's say, a reactive transport kind of problem. It's a really, really interesting field to study how the structure is dynamic. And we're hoping to be able to apply what we've learned right now from just static porous media that have different levels of complexity and apply to see how things are evolving in time. And so we're working specifically on that right now, both in the context of bioclogging and rock dissolution.
Yeah, that's great. I think hopefully in the future, this is something people going to be learning in the soil physics textbook, because assuming average matrix flow seems is not has some limitations in understand what's really happening in the nature.
Yes.
Thank you so much.
You're putting it very kindly. It has some limitations. It's wrong.
All right. Great, thank you.
Okay, thank you, Professor Morales. Thank you for your clear explanation. And I think I have to stop here, and it's the end of the talk time, I think. So thanks again for Professor Morales, your excellent talk, and thank Professor Zhang. And I hope we can meet her in the future in USA or in China after the pandemic, I think.
No, thank you both so much for the generous invitation, and EEH for hosting.
Thank you.
The seminar series. Really appreciate it. If there are any additional questions that come up, please feel free to email me. I'm sure the email can be accessed through the journal itself, but it's also listed at the bottom of my slides if you'd like to go back to the recording.
Okay, I will keep in touch with you all after this talk.
Excellent.
Thank you.
All right.
Thank you again.
Have a good day.
Bye.
Yeah, bye.
Have a good day.
Have a good night.
Bye.
Yeah.
See you.